Next we're going to hear from Dr. Ray Greek of Americans for Medical Advancement, which is a not-for-profit organisation advocating science-based medical research. Firstly though, I just want to run through a couple of slides from Dr. Greek's book, What Will We Do If We Don't Experiment on Animals? This first slide reads, looking just at the absorption process, the A in ADMET illustrates the enormous challenges posed by the ADMET problem. The way a drug can enter the body and the body's tissue can be complicated and varied. In order to get to the target, a drug must cross several membranes. First, it must pass from the environment to the interstitial fluid. This can be done by swallowing the drug and having it enter the interstitial fluid of the lining of the stomach or intestines. From the interstitial fluid, the chemical must cross a membrane to enter the bloodstream, and likewise, in order to exit from the blood to the target tissue, it must cross another membrane. Once in the interstitial fluid of the tissue it needs to penetrate, the drug must then cross a membrane to enter the cell. Scientists must study the chemical to understand what will happen at each of these membranes. This slide reads, and this is also from What Will We Do When We Don't Experiment on Animals. There are many factors that lead to a successful drug, but of particular importance is the way in which the human body absorbs and metabolizes the drug. This area of drug development, known as pharmacokinetics and pharmacodynamics, defines the drug's absorption, distribution, metabolism, and excretion, ADMET characteristics. Inappropriate ADMET parameters can lead to up to 40% of drug candidates failing to make it past the first human studies, phase 1 studies that is. For me, this figure of 40%, which can also be heard in this BBC Radio 4 programme.
Now we know that a lot of drug candidates fail to get anywhere near the marketplace, and I assume that quite a lot of them fail at the phase 1 test, don't they?
Oh, absolutely. I've been involved with drugs where after a single dose we stopped the trial. We would stop a drug about 40% of the time.
40% of the time?
Is the most damning evidence against the scientific validity of testing drugs and chemicals on animals. Why is this? Well, unlike the 92% of drugs that fail after all 3 clinical trials, this 40% failure rate is not on the grounds of efficacy, but purely on the grounds of toxicity. Phase 1 clinical trials are the first in human trials and only exist to ascertain the toxicity of a substance. If the animal tests were in any way accurate, you simply wouldn't get a 40% failure rate. This is because the animal tests would have obviously predicted the toxicity beforehand. Anyway, before we hear what Dr. Greek has to say in person, you can see this slide in front of you here. This is from a talk or a discussion between Dr. Hicks and Dr. Greek. Unfortunately there weren't any cameras there, but you can see the transcript on Dr. Greek's website. I only want to bring this up because it's the only thing really about the E in ADMET. I suppose they always look at the E but don't mention it much, but this is quite a graphic illustration of why animals can mislead you. They're talking about the discovery of penicillin, and someone asks about some question about it. Dr. Hicks' response to the question is as follows. I mean, it's a loaded question. First of all, the whole thing about penicillin. I can't answer that, your initial opening statement about penicillin, because I don't know the history of that. I don't know the detailed history of it. Dr. Greek, he jumps in. Actually, let me jump in. Penicillin was tested on a rabbit in 1928 by Fleming. It excreted penicillin so rapidly from its kidneys that Fleming put it back on the shelf.
This is a study published in 1990 where 6 drugs that were in clinical trials were studied for their side effects, and then people retrospectively looked back at the animals that were tested to see how everybody compared. And what they found was that animals and humans shared 22 side effects, and we call those true positives. Animals incorrectly identified 48 side effects that in fact did not occur in humans, and the animals missed 20 side effects that in fact did occur in humans. Now, if we use our little box down here in the right-hand corner, we're going to find that the sensitivity of that test is about the same as flipping a coin. And we're going to find that the positive predictive value of that test is 31%. Again, not a number that I would want to hang my life on. This is not my slide. This is a slide from Mark Levin, the former CEO of Millennium Pharmaceuticals. He showed this data at a drug discovery meeting in 2001 in Boston. And his company looked at 28 potential drugs and tested them all on rats to see if it damaged the rat's liver. That's what hepatotoxic means, liver toxic. And in rats, 17 of the drugs were safe and 11 damaged the liver. Levin said, let's test pretty much all of them on humans and just see what happens. Very small doses, they weren't going to kill anybody. And what they found was that of the 11 drugs that were toxic in rats, 6 of them were safe in humans. And of the 17 that were safe in rats, 6 of those were toxic in humans. Now what Levin concluded from this, and this isn't, well, it is my conclusion, but it was Levin's conclusion first, is that the rat data was about as accurate as a coin toss. For those of you who don't have a science background in the audience, this is a type of graph that we call a scattergram. And in this study that was published in 2002, the authors compared animal bioavailability of drugs, which is on the x-axis, with human bioavailability of those drugs, which is on the y-axis. And they looked at primates, rodents, and dogs and compared that data with humans. And as you can see, there is absolutely no correlation whatsoever in this data. This is a meeting that I attended 1 year ago this week that was sponsored by the CBI, and it was a really neat meeting. It was a closed meeting, and I only got invited through a fluke. But it was comprised of people from the pharmaceutical industry, and the NIH, and the FDA. And like I said, it was a closed meeting, and it was pretty neat because pretty much everybody who was in the audience was also a speaker, again, except for me. And the premise of this meeting was that animal models are not predictive for humans. Okay, that was the given. And it was neat because the pharmaceutical companies came together and shared data. So Pfizer's up there saying, we don't have anything that predicts what a drug is going to do in humans. And it's predictive ADMET. A stands for absorption, D for distribution, and so forth. And these are the parameters that we try to use animals to determine. Okay, ADMET is what it's called for short. So Pfizer gets up and says, here's what we're doing, it doesn't work, but, does anybody else have any ideas? And then the scientists from Merck get up there and say, we know animal data doesn't work, we're trying this computer method, and so on, and so on, and so on. But it really kind of floored me after 10 years of going around the country and saying the animal model is not predictive, to have representatives of NIH, FDA, and, the big pharma up there agreeing with me.
Next we will hear from Dr. Margaret Clotworthy, who is the Science Director of the Safer Medicines Trust. Dr. Margaret Clotworthy, on the 26th of November 2009, helped organise and present a groundbreaking international conference entitled Speed and Safety in Drug Discovery. The proceedings of the Speed and Safety in Drug Discovery Conference can be read in this ATLA journal. You can also listen to the conference if you click this link on the Safer Medicines Trust's website. The following clip, however, is taken from an interview Dr. Margaret Clotworthy did on a programme called On the Edge. This was shown on Sky Channel 200 on March the 18th, 2010. The whole interview can be seen on the Safer Medicines Trust website.
Yeah, well, somebody, Brian in South Wales, is saying that he's a software engineer and a vegetarian, and he says, computers, garbage in, garbage out.
That's true. That's true. If you don't put in good quality data, you won't get out good quality data. That's absolutely true. That's why you constantly update the information in your models with as much information as you have. But also, you wouldn't just rely on computer models. You would look at tests in basic cell cultures, tests in human tissues, in microfluidics chips, which link different sorts of tissues.
Now, microfluidics chips, these are the little plastic chips with pits in them, and in the pits lives human cells, like there might be liver cells or heart cells or kidney cells.
Yes, or a combination of all of them.
Okay, and you create an environment around them which simulates the human environment, the inside the body of the human.
Yes.
And then you dose it with this drug.
Yes.
So I don't understand. What happens then?
Well,
what can you possibly tell?
Well, there are a number of ways of analysing the results from such a study. You can look microscopically using different stains, etc., to see whether any damage has been done to the tissue, whether they've decided to produce more of a certain receptor. You can do biochemical analysis to find out whether they're sending out distress signals, so different hormones that say, that would recruit inflammatory cells, etc. You can also look genetically to see whether certain genes are being upregulated, turned on or turned off. And then you can basically compare those results to the results from drugs whose effects are already known, for example. And in that way you can tell what the drug is likely to do. And because these chips would contain liver, so you're looking at metabolites, how the cells break down the drug you're interested in, or drugs if you're testing more than one. And you can look at the effects of those breakdown products on the other tissues in question as well.
Explain metabolites.
Oh, well, when you take in a drug, or indeed food, you digest the food, essentially, and the liver is the organ responsible for digesting, so to speak, drugs and other potentially toxic molecules. Sometimes it will produce... So it basically splits the molecules up into different bits and pieces so that it can be excreted, so the body can get rid of it in the urine or feces. And sometimes those breakdown products, those digest metabolites, will be active. They'll be more active than the drug in some cases. It will bind to the receptor or inhibit the enzyme that it's destined to interact with better. Or it might target a different organ, for example, than the one that is intended. So it's really important to look at that, and that's where technologies such as microdosing actually come in.
Okay, well, let's talk about microdosing, because this is with human volunteers, presumably.
Yes.
But well, you said, and correct me if I'm wrong, but that those people at Northwick Park who became very seriously ill were given doses that were 500 times smaller than the doses that have been given to monkeys that didn't respond adversely at all.
Yes.
So microdosing is how many times smaller than that?
It would be, in the case of the Northwick Park drug, it would be, because that's a large molecule drug, it was an antibody, it would be about a 60th or a 70th of the amount that they were given. I'm not saying it would be good for you at a 70th of the amount, but it probably wouldn't have caused quite such drastic effects. So what happens is you label the drug radioactively, but because you're using such a small amount of drug...
So, hold on, you're giving this to people, you're giving them radiation. That's
tiny,
but tiny.
Tiny amounts. You get radiation walking outside in the street or flying.
Well, from the sun?
Yes.
Well, okay, yes, you do, but you don't want any added radiation, do you? I mean, I know when you get your teeth x-rayed, you get radiation and so on, but...
Yes, different sorts of radiation. You get really, really a
so
tiny amount.
So you tag this tiny amount of drug.
Yes.
And then you can find out where it goes by scanning people.
Well, yeah, PET scanners, such as you have in hospitals, already diagnostically they're used to look at the body, so you can see where the drug goes. You can take blood samples and urine samples and find out what those breakdown products, those metabolites are.
Okay.
And because you're using such tiny amounts and you're using the species of interest, i.e. humans, you can find out what will happen. And studies have shown that that scales up to pharmacological doses at least 80% of the time.
Well, it sounds to me a bit like homeopathy, where you have these incredibly watered-down amounts. And in the film that we're going to show after this programme, they actually say, and again, correct me if I'm wrong, that they can detect a litre's worth of any drug in all the oceans of the world.
Well,
that's homeopathic, isn't it? Surely.
Well, the difference with microdosing is, okay, you're using AMS, which is, the most sensitive
So you have
detector
to
in the world.
explain what
Sorry, accelerated mass spectroscopy.
mass spectroscopy.
Yes, so it's basically a really, really sensitive way of picking up those tagged
This is
molecules.
a thing that produces those little bars
Blips,
that say this is this element and this is this element. Is that right?
That's right. Although we're not looking at elements, we'd be looking at, a signature which would identify a drug or a breakdown product of the drug. So this is this part of the molecule, that's that part of the molecule that's broken off, that's been digested.
Okay.
So it's incredibly sensitive. You wouldn't quite be using it at the limits of its detection. And also, these drug amounts are designed to be so small that they don't have any effect because they're really tiny. So you're just looking in a safe way at what happens to the drug. So it's not... I understand, why it sounds like homeopathy, but it's not. It's not designed to have any effect.
In this last part of pharmacokinetics and metabolism, we will hear from Dr. Katya Tsaioun, who is the president of Apredica. Dr. Katya Tsaioun's presentation, which has been slightly shortened here, was given at the Speed and Safety in Drug Discovery conference just mentioned. Dr. Katya is a scientific advisor to the Safer Medicines Trust. She also appears briefly in another episode of Toxicity Tests called Genotoxicity.
First of all, I'd like to thank the organizers and Margaret in particular for, she's not here, for inviting me. This has been a very exciting day. And I have to say that I actually, because I was planning to fly in this morning from the US, that's why Margaret put me in the afternoon session, but we really are focused on in vitro models, so I really belong more in the previous section or even maybe the first one. But with that in mind, I'll just tell you a little bit about ADMET. I'm sure there's very sophisticated people in the audience, so a great majority of you know what ADMET is. And again, it's been really exciting to hear what other companies and groups have been working with on very cutting-edge stuff. What myself and my company does is really more bread and butter ADMET. So what we bring to the table is really more an approach to this. And for big pharma, what we do has become really the commonplace. However, there is a lot of academic groups and spin-offs from academia and small companies that are not aware that this actually exists, if you can believe it, because you are very far out there with your technologies. There's actually whole groups of people who want to do drug discovery, who have very exciting targets and molecules, but don't know that there are some pitfalls that they should avoid. So I'll go very quickly over the in vivo and in vitro approaches to ADMET, which of course everybody knows here, and the value of effective ADMET strategy. And then I'll quickly go over a couple of new in vitro ADMET profiling tools that we have developed in partnership with other groups, both academic and commercial. And I'll show you a bit of a piece of entertainment at the end if you survive the first part of my talk. So this is a problem that actually just a few weeks ago a client of ours has presented to us. They've tried the rat and mouse PK. The drug wasn't orally bioavailable. The CACO2 model didn't work. They actually, in spite of that, they did put the drug into clinical trials because they were, for whatever reason, and actually confessed to us that just looking at the rodent data, we would never have selected the compound to go forward. And I think that this is a very compelling example of how we use the animal data and how sometimes it's not. This is a real stuff that's happening right now. They're trying to figure out what to do with follow-up compounds and how to screen them, because essentially they have to put it straight into man or something like that. So solutions, and there's certainly very exciting stuff that I heard this morning and also know from other groups in the US that people are working on stem cells approaches on tissues, and however we still don't have the one solution. So let me take you back a little bit and see where the ADMET, how the ADMET field evolved. So the accepted in vivo ADME assays, obviously it's biodistribution, absorption through GI or BBB. Most of it historically has been done with radioactively labeled compounds. Later in development, which is, it's an expensive proposition, requires a lot of compounds, so you can't really do screening with any of those assays. And finally, toxicity, it's so-called gold standard. So how are these assays used? Obviously later in development. However, the PK/PD assays are frequently used for lead optimization to see whether the drug is bioavailable. But as we saw in one example already, that's not always predictive. So what are the problems with this? Well, with one paper that I saw, the human toxicity prediction is somewhere on the order of 55-60%, which is just a little bit better than flipping the coin. However, this is the best that we've got. So the mechanisms of toxicity could be quite different in an animal compared to human, as we all know. So what are the results? This is the data from 1993, and close to 40% of drugs failed for simple DMPK reasons in the clinic. So about that time, with advent of high-throughput screening and miniaturization, there was a lot of effort to bring in and develop the early ADME assays. Something that at that time was really cutting edge, that's now basically a background for all big pharma to screen, to do lead optimization on their compounds. Not necessarily on every compound, but certainly on compounds that have shown in vitro efficacy and sometimes in vivo efficacy. And depending on the therapeutic area, you certainly pick and choose in terms of priority and assign to different assays different weight and importance. So that's, again, with the CYP450 enzymes expression and such. These were significant achievements and milestones in the pharmaceutical industry, which is now considered the basic stuff that everybody should do. But this is probably why I got so fascinated. About that time, I actually started doing ADME at GPC Biotech, where I was working at the time. It was about 95, 96. And I went to my first ADME conference, and I was really fascinated by the wealth of information that you can get early on when on the actual human enzymes and human cells sometimes. So these are some. So I sort of divide these assays into routine ADME assays and more for advanced profiling, because you certainly don't necessarily need to know how each drug, by which transporter each drug is transported in the GI tract at the beginning. However, as you move forward in the development, this is something that even FDA now has a recommendation to do. And when FDA recommends something, people usually try to do it. Otherwise, there might be consequences. So some of these, so I would classify a lot of the assays that were presented this morning more in the profiling stage, where you're working not on thousands of compounds, with a few exceptions, but majority of the technologies there were made for dozens of compounds or so. And I may be wrong, depending on what assays you're talking about. So, and finally, the in vitro DDI package is something that the guidance from FDA, which I'm sure you're familiar with, that came out in September of 2006, that defines pretty precisely how you should go about CYP450 inhibition induction and PGP testing. So how we use them now? So the developability, which is a term that's frequently used. So the compound is binding to the receptor. Well, is it going to be druggable? Is it going to be soluble, and such things? So it allows us to provide actionable information early, not only to kill compounds, but also to sort of have a smarter approach and look at the structure and figure out which spinach on the molecule are creating certain problems, or which ones are responsible for the biological effect. So, and certainly I've seen some presentations this morning about from the future in terms of whole body metabolism prediction or virtual patient and such, which is very, very exciting. That's certainly where the field is moving, not as fast as most of us would like, but So what was the impact of starting to use all these technologies? And obviously, you probably am preaching to the choir, so everybody saw that paper, where the advent of early ADME really has brought in the failures in the clinic due to DMPK reasons to down to 7% from under 40%. So what ADME can do to lead optimization is really when it's done at the same time with efficacy to select the preclinical candidates, it really could move your IND and subsequently your NDA to faster times, which means that you hopefully will have more time on the market, because we're talking about 20 years of patent life, which everybody is racing against. But still, there is. This is from the FDA website, and these are the list of drugs that have different issues in terms of toxicology, which have come up after release in the public. And granted, in many cases it's impossible to predict any of those, because a lot of those fall under the idiosyncratic toxicity label, which means that you can't possibly conduct a clinical trial with 20,000 patients. However, some of these have common mechanisms of toxicity that I'm going to talk about. It's not getting degraded in the cell, so the fluorescence goes up. So tamoxifen actually has phospholipidosis as a problem, so we see this in this assay, and coinciding with the toxicity, which is in the bottom panel, whereas in acetaminophen we don't see this effect. We also have a number of partners. One of them is Chantest. We like to partner with companies that are specializing in a particular area. So they're real experts in electrophysiology. I'm sure you're familiar with them. So we've developed this one assay. It's essentially a screening assay for CYP450 for drug-drug interaction and cardiac toxicity. Green Screen is another. Gentronics is another partner that we have. So we offer the Green Screen for early genotoxicity screening, and I'm sure most of you are familiar. It's a UK company, but it's much more predictive than AIMS. It's using mammalian cells, and is possible to do in very small amount of compounds. HepRG is a human hepatic cell line that has all the metabolic activity as primary human hepatocytes. It's out of INSERM and is distributed by Biopredict, is available on licensing basis. So these are some of the data that was reproduced in our lab, which shows metabolic profile of a couple of compounds, aflatoxin B1 and acetaminophen. On the left is HepRG cells, on the right is human primary hepatocytes, and as you can see, most of the metabolites actually are quite identical here. So that's actually very exciting. So what you also could do, not only metabolism and induction studies in the cell line, which is obviously as a cell line, it's readily available. You don't have to wait for people to die to get the cells. It also could help in toxicity prediction, because in this particular case, aflatoxin B1 needs metabolic activation to become toxic. So you can see that in HepG2 cell lines, which is a pink line, you don't see much toxicity going on. However, in HepRG cell line, you do see the toxicity because it's metabolically activated, because the cells have all the metabolic enzymes. As opposed to that, in the amiodarone is toxic by itself. It doesn't need any metabolic activation, and you can see no difference. So again, this is a model. A model will tell you what the model can tell you. So it's very important to understand what your model does. So these are some of the technologies that we are working on in the background and trying to evaluate. So human blood-brain barrier model commercialization. We have a collaboration with MIT Draper Labs on hepatocyte technology, with some technology coming from the University of Singapore, and one startup that we're looking at, fast metabolite ID with fast biomimetic synthesis. So there's, in summary, there is no easy solutions to pharma problems, as we all know. We certainly have enough data to show that ADME has proven its value for drug development. And the next goal is, of course, to develop toxicity models that are relevant to human, which, of course, is why we're all here. And I would like to thank again Margaret and everybody for this. And this is acknowledgement of certainly the top scientists in my company who have generated a lot of that data. The Buzz Brown Chan test, we had a lot of good discussions with Christophe Chesnay from Biopredict, John Nicholson, who is the chairman of Gentronics, and Aloise, who is an academic professor from ICEA in Paris. And thank you very much again.